Imagine you're trying to power up some kind of complex machine, maybe like the Millennium Falcon. But instead of adding fuel, you're just pulling out a dampener.
That's pretty much it. That's the fundamental truth about your morning coffee.
So am I actually getting this massive jolt of energy, like downing a power-up potion? Or am I just like casting a really effective counterspell on the signals telling me to slow down?
It's definitely the latter, a counterspell. And it's kind of the ultimate paradox, isn't it? Caffeine doesn't create energy. It just cleverly disables the body's natural brake.
Okay.
And understanding that mechanism is really the first step. If you want to appreciate the extreme chemical and biological efforts scientists go through to take that effect away, to create the perfect cup of decaf.
Welcome to the deep dive. Today we are really unpacking the whole story of the coffee bean. We're starting deep inside your brain, then tracing the path through some, let's say, controversial industrial vats.
Yes, some interesting history there.
And finally landing on this, groundbreaking genomic discovery. Something found in a wild, endangered species that could genuinely change the future of how we brew coffee.
Absolutely. And this dive is really custom built for you, the learner, who wants the real story. Not just if decaf is safe, but how science is racing to engineer the ultimate, naturally buzz-free cup.
All right, let's unpack this.
Let's do it.
Okay, let's start with that alertness engine then. If caffeine isn't gasoline for the brain, what is it doing? I think I remember the keys. This tiny molecule builds up sleep pressure, adenosine.
Exactly. Adenosine, that's the one. Think of it as your brain's sleepiness accountant. The longer you're awake, the more adenosine accumulates.
Okay.
And when it binds to certain receptors in your brain, it basically signals, hey, time to slow down, promoting that feeling of sleep pressure, or what scientists call the homeostatic sleep drive.
It's like the body's gentle nudge saying, maybe take a break.
Precisely, a persistent reminder. Now, this is where caffeine pulls its trick, the molecular mimicry act.
Right, the counterspell. How exactly does it fool the brain?
Well, caffeine's structure is incredibly similar to adenosine, almost identical in key parts. So it acts as what we call a competitive antagonist, meaning it rushes in and binds to those same receptors, especially the A eurotype, before adenosine can get there. But here's the crucial part. Caffeine doesn't activate the receptor. It just sits there, occupying the parking spot.
So it's like putting duct tape over the sleepiness button. The signal's sent, but the button doesn't work.
That's a great way to put it. The signal is blocked. The message can't get through.
Okay, so we've basically disabled the brake. But that feeling of feeling more alert, faster, maybe a little jittery, that has to come from somewhere else, right? It's not just the absence of sleepiness.
Absolutely right. When caffeine blocks those adenosine receptors, it has knock-on effects. It indirectly modulates other systems. And that's where the rush comes from. Blocking adenosine actually enhances dopamine activity. Research shows a clear link between how alert you feel and increased availability of dopamine receptors, specifically in the striatum part of the brain.
Okay, dopamine. That makes sense for feeling good and motivated.
Yeah, and plus, you get a boost to the sympathetic nervous system. Caffeine encourages the release of norepinephrine and other catecholamines. That's your body's kind of fight or flight chemical boost, kicking in increased heart rate, vigilance.
Wow. So it's a 1-2 punch. You remove the brake by blocking adenosine, and you nudge the accelerator with dopamine and norepinephrine.
You got it. But, it gets even more complex.
Yeah.
Because that feeling often lasts much longer than you'd expect just from the initial caffeine hit. There's a second act.
A second act.
We need to talk about paraxanthine. After you drink coffee, your liver gets to work breaking down the caffeine into several metabolites. Paraxanthine is the main one in humans.
Okay.
And what's really fascinating is that paraxanthine often reaches higher levels in your bloodstream than the original caffeine itself. And it clears out more slowly.
So it sort of takes over the job.
It picks up the alertness baton exactly. It can continue the effect, potentially extending that feeling of being switched on for several more hours.
Ah. It's like the quiet sidekick that keeps the superhero's powers going after the main event.
Kinda, yeah.
Now, if the basic mechanism is the same, why do some people feel amazing after a double espresso, almost superhuman, while others just feel anxious and shaky?
That's where your personal biology, your genetics comes in. It largely comes down to one specific gene, CYP1A2.
CYP1A2.
That's the one. This gene basically determines how fast your liver metabolizes caffeine. If you have the AA version, the AA genotype, you're a fast metabolizer. meaning you clear caffeine about 1.6 times faster than others. You tend to get the benefits, the alertness, the focus with fewer downsides.
And the other type?
If you have the CC genotype, you're a slow metabolizer. Caffeine lingers much longer in your system. This increases the risk of feeling jittery, anxious, maybe having trouble sleeping, especially if you consume a lot.
Wow. So my own DNA sets the speed limit for my coffee intake. That explains a lot.
It really does. It's personalized chemistry.
Okay, so if we're talking optimization, what about timing? What's the best time to have that coffee, considering our natural body clock?
Yeah, the research on this is pretty cool and practical. Your body has a natural surge of cortisol, the alertness hormone, shortly after you wake up. It's called the cortisol awakening response, or CRR.
Right, I feel that sometimes.
Well, to avoid piling caffeine stimulation on top of that natural peak, which can actually blunt the cortisol response over time or just make you feel overly wired,
Yeah.
the recommendation is to wait about 90 minutes after waking before having your first coffee.
90 minutes. Wow. Okay, that's a potential game changer for my mornings.
Give it a try. It lets the natural cortisol do its thing first, then the caffeine comes in to extend the alertness rather than fighting with it.
And I guess there's a flip side. A warning for evening coffee drinkers.
Oh, definitely. A big one. The circadian warning is pretty stark. Consuming the caffeine equivalent of a double espresso, say just 3 hours before your normal bedtime, can actually delay your body's natural melatonin rhythm by about 40 minutes.
40 minutes? That's significant.
It is. You're literally pushing your internal clock back, making it that much harder to fall asleep, and potentially messing up your sleep cycle.
Okay, so we've figured out how caffeine puts the buzz in. Now let's talk about the flip side. The incredible, sometimes controversial, effort involved in stripping that buzz out. Decaffeination sounds like this. Messy history, right? A mix of chemistry and physics.
It really is, and the fundamental challenge is huge. You're trying to remove one tiny molecule, caffeine, from the green coffee bean.
While leaving everything else.
Exactly. While keeping intact over 1,000 other different, often very volatile compounds that create the complex flavor and aroma that we love in coffee. It's a delicate operation.
So let's get the elephant in the room out of the way first. Chemical decaffeination. When people worry about decaf being bad for them, they're usually thinking about solvents, right? Like methylene chloride.
That's often the one people mention, methylene chloride, or MC. And look, it's still one of the most widely used methods, mainly because it's very effective. It removes about 96 to 97% of the caffeine.
How does it work? Do they just soak the beans in it?
There are direct and indirect methods. Sometimes beans are steamed and rinsed directly with MC. Other times, beans are soaked in water, the caffeine and solids move into the water, then that water extract is treated with MC to remove the caffeine, and finally, the decaffeinated water extract is added back to the beans.
But MC, isn't that used in, like, industrial stuff? Paint strippers.
It is. Methylene chloride is used as an industrial solvent, including in paint strippers and degreasers. Now, it's important to say the FDA has very strict limits. Residual solvent must be below 10 parts per million, which is tiny.
Still, the association isn't great.
No, that connection to industrial chemicals is definitely unsettling for a lot of consumers, even with the regulations.
Okay, is there another chemical option that maybe sounds a bit friendlier?
Well, there's ethyl acetate, or EA. This one is often marketed as natural, or the sugarcane process, because EA is a chemical found naturally in fruits and ripening vegetables and can be derived from fermenting sugarcane.
Ah, so it's actually natural then.
Well, yes and no. While it occurs naturally, the EA used for large-scale commercial decaffeination is typically synthesized industrially. It's chemically identical, but it's made in a lab or factory.
Gotcha. And how effective is it? Any downsides?
It's similarly efficient to MC, around 96–97% caffeine removal. The challenge with EA is that it can sometimes be harder to remove completely from the beans. If you've ever had a decaf that had a slightly sweet, almost varnish-like, or fruity but off aftertaste, that might be residual EA.
Right. Okay, so those are the main chemical players. Let's pivot to the methods that avoid solvents altogether, the ones that rely more on physics. The Swiss water process is probably the most famous one, right?
Definitely the most well-known solvent-free method. And the Swiss water process is really quite ingenious because it relies purely on diffusion and solubility, not on added chemicals.
How does that work?
Okay, so they start by taking a batch of green coffee beans and soaking them in very hot water. This extracts everything caffeine, oils, flavor compounds into the water. This creates what they call green coffee extract, or GCE.
Right, so it's a super concentrated coffee water.
Exactly. Then they take that GCE and pass it through special activated carbon filters. These filters are designed to specifically trap the larger caffeine molecules while letting the smaller oil and flavor molecules pass through.
Okay, so now the GPE has flavor, but no caffeine.
Precisely. It's flavor-rich but caffeine-poor. And now comes the clever part. They take a new batch of green coffee beans and they soak those beans in this caffeine-free GCE.
Ah, and because the GCE is already saturated with all the flavor stuff.
The flavor compounds mostly stay in the new beans. But the caffeine, since there's lots of caffeine in the beans and none in the surrounding GCE, the caffeine naturally migrates out of the beans and into the liquid, trying to reach equilibrium.
Driven purely by the concentration difference. Wow.
Purely by diffusion. No solvents needed. They keep refreshing the GCE through the carbon filters. It's highly effective. They claim 99.9% caffeine removal, and it's highly valued for being clean, sustainable, and generally preserving flavor well.
That's pretty cool. And then there's the really high-tech one involving gas.
Right. The supercritical co-Euro method. This is probably the most technologically advanced and often the most expensive.
Supercritical. What does that even mean?
It means they take carbon dioxide, co-Euro, which is usually a gas, and they subject it to high pressure and temperature above its critical point, around 90 degrees C and 250 times atmospheric pressure.
And what happens then?
It enters a weird state where it's neither just a liquid nor just a gas. It behaves like both. It can effuse through solids like a gas, but dissolve things like a liquid. And in the supercritical state, co-Euro becomes a remarkably selective solvent for caffeine.
Selective. So it grabs the caffeine but leaves the rest.
Pretty much. It's very good at dissolving the small caffeine molecules, but it tends to leave the larger, more complex flavor and aroma molecules behind in the bean. It's like a precision tool.
Which means better flavor retention.
Generally, yes. That's why it's often used for high-end specialty decafs. The big downside is the energy required to create and maintain those supercritical conditions. It needs specialized, expensive equipment.
Okay, that makes sense. Now, before we get to the genetics,
Yeah.
you mentioned bacteria that eat caffeine. That sounds like something from a sci-fi movie. Is that real?
Oh, it's absolutely real. It's still mostly in the research phase for commercial use, but scientists have identified and studied bacteria, like certain strains of Pseudomonas, that have evolved enzymes to literally break down caffeine. They use enzymes, specifically things called endomethylases, to sequentially chop bits off the caffeine molecule, turning it into theobromine, then other related compounds like xanthine, effectively dismantling it.
Wow.
One study, for instance, using Pseudomonas alcaligenes CFR 1708, showed it could completely degrade a fair amount of caffeine, like 1 gram per liter, in just 4 to 6 hours under the right lab conditions.
So maybe one day our decaf will be made by
Microbes, it's a possibility. It points towards potentially ultra-sustainable, highly specific biochemical processing methods for the future, a biological frontier for sure.
Okay, so as cool as high-tech gas chambers and caffeine-eating bacteria sound, the ultimate solution, right, the holy grail would be a coffee plant that just doesn't make caffeine in the first place. No stripping needed.
Exactly. That avoids all the processing, potential flavor loss, cost, environmental impact, everything. And this brings us to a really exciting genomic revelation, a huge development.
And this discovery centers on a specific kind of obscure coffee species.
Yes, a wild and unfortunately endangered species called Coffea humblosiana. It's native to the Comoro archipelago, islands off the coast of East Africa. And the key thing is, it's naturally caffeine-free.
Naturally caffeine-free. So scientists obviously wanted to know why. How did they figure it out?
Well, a major piece of the puzzle came from a paper published in the journal Scientific Reports. The lead author was Natalie Raharmala, working with Stefan Rambaud, Dominique Cruziat, and a whole team of colleagues.
Any notable affiliations there?
Yeah, it was a big collaboration involving researchers from places like the Centre National de Recherche Appliquée au Développement Rural in Madagascar, Ghent University in Belgium, and even Nestlé Research.
Okay, so what did this international team actually do?
They essentially assembled the genome sequence of C. humblosiana. They mapped out its genetic blueprint, and then they compared it side by side with the genome of a common cultivated coffee, Robusta, coffea caniphore, which obviously does produce caffeine.
And what did that comparison reveal? What was the big finding?
They found a missing piece, a crucial missing instruction in the genetic recipe book. See, in normal coffee plants, there's a whole pathway, a series of steps to build caffeine. The very last critical step is converting a precursor molecule called theobromine.
The stuff that's famously in chocolate.
That's the one, converting theobromine into caffeine. This final conversion is done by a specific enzyme, and the instructions to build that enzyme are contained in a gene called caffeine synthase, often abbreviated as DXMT.
Okay, the DXMT gene makes the enzyme that makes the caffeine. So they looked for this gene in the naturally caffeine-free C. humboldt, and what did they find?
They found nothing. The DXMT gene was completely absent on its chromosome 1. It wasn't just broken or mutated. The entire section of DNA containing the blueprint for that final caffeine-making enzyme was simply gone.
Gone, like deleted.
Likely lost sometime during its evolution, probably through a specific type of genetic error called illegitimate recombination. The instruction manual was missing the last page.
Wow. And did they double-check this, like, chemically?
They did. Biochemical tests on the leaves of C. humboldtiana confirmed it. They found the precursor, theobromine, was present. But there was absolutely zero caffeine. The production line stopped right before the final step because the machinery wasn't there.
That is incredibly specific and potentially game-changing, right?
Right.
A single missing gene is the whole reason.
It seems to be the primary reason, yes. And that finding is huge for the future of coffee. It provides a crystal-clear genomic target. Meaning, now that researchers understand the exact genetic difference, the absence of this DXMT gene, they have a roadmap. They could potentially use traditional breeding techniques, crossing C. humboldtiana with cultivated varieties, or maybe even modern genetic editing tools like CRISPR.
To basically delete or disable that same gene in regular coffee plants, like Arabica or Robusta.
Precisely. To develop new cultivated coffee varieties that are naturally caffeine-free right from the start, it could completely eliminate the need for all that post-harvest decaffeination processing we just talked about.
That would be a massive shift. Sustainable, potentially better tasting.
Absolutely. It's a sustainable alternative that tackles the issue at the source, the plant's own genetics.
Okay, we have journeyed from the tiny gaps between brain cells all the way to the genetic code of a rare wild coffee plant. Let's quickly recap the main takeaways from this deep dive.
Sounds good. So first, remember that caffeine's magic trick is molecular mimicry. It works primarily by blocking your brain's natural sleep pressure signal, adenosine. It's not fuel, it's a brake blocker.
Right. And how you experience that block break is highly personalized, thanks largely to your CYP1A2 gene, which dictates if you're a fast or slow metabolizer.
Exactly. Second takeaway. The quest for better decaf is this ongoing technological race. We're seeing a clear shift away from reliance on industrial solvents like methylene chloride,
The paint stripper chemical.
towards cleaner, purely physical methods that prioritize flavor and sustainability, like the solvent-free Swiss water process or that high-precision supercritical co-euro extraction.
Okay, blocker, better processing. What's the third?
And finally, perhaps the most profound point is the biological solution. The discovery in coffee humboldtiana that it's naturally caffeine-free because it completely lost that single crucial DXMT synthesis gene gives us the genetic blueprint.
The key to potentially engineering truly sustainable, naturally decaffeinated coffee right from the plant itself.
That's the ultimate goal.
It really feels like we're moving towards a future where our relationship with coffee becomes incredibly tailored, influenced by our own DNA and by science's growing ability to tweak the bean itself.
It does, which leads to a pretty provocative final thought for you to mull over.
Okay, let's hear it.
Given that researchers now know the specific gene deletion responsible for making a coffee bean caffeine-free, will the next big trend in coffee be hyper-customization? Imagine a future where the caffeine level in your cup isn't just regular or decaf, but maybe it's matched precisely to your personal DNA profile based on your CYP1A2 results.
Whoa. So you walk into a coffee shop, they scan your genetic profile and brew a cup with the exact caffeine kick optimized just for you.
Could happen. Will your future barista ask for your genotype before pulling your morning espresso? Something to think about.
Yeah.